It will change. Can you check?
Piraj, please replay it again so that others will also join now.
Sunil sir, over to you, sir.
Okay. Good evening, one and all. I welcome you all to the one-day international webinar on role of biopharmaceutics in drug discovery and development. So, let us start the today's session. So, guest speaker of this session, Om Perumal, our director Dr. Kumar V.S. Nemmani, principal Dr. K. Prasad, pharma fraternity from various colleges and all participants. On behalf of Sri Vishnu College of Pharmacy, we welcome you to the one-day international webinar. On this note, I would like you to introduce to our director and principal to the gathering. Dr. Kumar V.S. Nemmani, Director, Sri Vishnu College of Pharmacy since 2018, holds B.Pharm and M.Pharm Pharmacology degree from Andhra University, Visakhapatnam. He has completed a PhD degree from Naipar Mohali and subsequently postdoctoral fellowship from McGill University, Montreal, Canada. Dr. Kumar V.S. Nemmani has over 14 years of industrial research experience on drug discovery and development and managerial experience. Introducing our principal, Dr. K. Prasad, completed his B.Pharm, M.Pharm and PhD from Andhra University and has worked in various teaching designations, including the prestigious HKE College, Gulbarga, and now serving as the principal for college since 2011. He has wide research experience and has managed several DPT and ACT research grants to his credit. Now I request Dr. Kumar V.S. Nemmani, Director, Sri Vishnu College of Pharmacy, to give a welcome note.
Good afternoon, everyone. I welcome each one of you for this international webinar. In fact, the college has organized about 20 webinars from March 20th to May 15th. But we didn't go live during those webinars. And from today is the first one where we are going live the webinar series. I'm very glad that you all joined this webinar series on this COVID pandemic. I must thank each one of you for joining this webinar. I must welcome you all for this webinar series on behalf of our leadership team, that is Dr. K.V. Vishnuraj Garu, Chairman, Dr. Sri Ravichandran Rajgopal, Vice Chairman, and our Principal, Dr. K. Prasad Garu, and on behalf of every staff member of Sri Vishnu College of Pharmacy. At this juncture, I also like to thank each and every principal and senior staff of all other colleges who are continuously supporting the webinar series. So at this juncture, I'm very glad for two reasons. The one reason is the number of registrations that are there for this international webinar, one-day international webinar, reaching about 1,500. So which is a good number in just 2 days of information to the media. And I'm also very happy for the other reason because I'm going to see my friend talking. Probably I'm going to see his talk after 2 decades nearly, I guess, and to see him in live action. And that another reason I'm very happy is the title that he has chosen, role of biopharmaceutics in drug discovery and development, which is relevant for the most of the pharmacists. So whether it is a PharmD program or whether it is a B.Pharm program, and in many ways it touches upon a PG program. So the title was aptly chosen by Dr. Om Perumal, and is the professor in pharmaceutics, and is a good friend of mine. I'm very proud of him for all his achievements. As the name says, Om Perumal, and he has all tick marks with respect to the piece. Whether it is publications, he has more than 100 publications. Whether it is patents, it's almost more than 30 patents and about 6 issued patents. Whether it is presentations, there also has a tremendous number. I'm really proud of you, Dr. Permal, and thank you for accepting our invitation readily. I know you do a lot of activities at a time, and with your talk today, many of the participants will be enlightened with their knowledge. So I also requested Dr. Perumal at the end to add a few points how to get into research, especially if you want to do MS program in US or graduation program in US, how to get into those programs. So I must thank Dr. Perumal for accepting this invitation and going to enlighten all of us. So, so with this note, I welcome once again all of you who have joined just now. Hope this webinar series will be a very useful and meaningful and interesting to all of you. So thank you for participation. So with this note, I will pass on to Dr. Sunil again.
Yes, sir. So let me introduce our today's speaker. Dr. Om Perumal is professor and a department head for pharmaceutical sciences in the College of Pharmacy and Allied Health Professions at South Dakota State University. And he completed his B.Pharm from Dr. MGR University, M.Pharm from Birla Institute of Technology, India, and a PhD in pharmaceutics from NIPER. Dr. Perumal completed his postdoctoral training at the University of Kentucky and Wayne State University. So he has 15 years of teaching and research experience, and he is the coordinator for the PhD program in pharmaceutical sciences. He served as the director of an interinstitutional translational cancer research center between SDSU and Sanford Research. The technologies developed by the department faculty has resulted in 30 patent applications and 6 issued patents and 7 technology development licenses with various startup companies. Dr. Perumal has published over 50 peer-reviewed papers, several book chapters, and has made over 100 presentations in various national and international meetings. And as a principal investigator and co-investigator, Dr. Perumal has received funding to the tune of $4 million. His group has developed and licensed several new drug delivery technologies. Dr. Perumal is the recipient of the prestigious Patricia Walker Skin Cancer Research Award from the Skin Cancer Foundation, and he is also a recipient of Butler Award for Excellence in Research, and he is also a recipient of Intellectual Property and Commercialization Award from the South Dakota State University. So with this introduction, I request our speaker, Dr. Om Perumal, to begin his talk. Over to you, sir.
Thank you, Dr. Sunil. I think, okay, I'm able to share my screen. Okay, I think, yeah, now I could. Yeah. So can you see the screen? Thumbs up, please, if you can. See
Yes,
the screen?
yes.
Okay.
Yeah.
So again, good afternoon to all of you. First of all, again, thanks to Dr. Sunil for the kind words and the kind introduction. And again, I would like to first start off thanking the college administration, the chairman, vice chairman, the principal for the invitation and the organizing this one-day international webinar. And last but not the least, my good friend, Dr. Nimani. Again, I'm equally excited to, see Nimani after a long time. I mean, more in a professional setting, but although personally we have been chatting for the probably last month or so after a long time. So I know he had gone to the industry and then came back to academia. So definitely experience from industry and more applied. And the other thing I was saying, before I start, maybe just a few words about the college. I saw the video before we started. It was very impressive. In fact, it made me, think about going back to college. I mean, with all the facilities that we have, it's a beautiful campus. Again, once again, thank you for the invitation. And also, I'm humbled by the response. And Dr. Iman is talking about 1,500 or so registrations. I'm really humbled by the response. And hopefully justify the expectation and try to do my best. The goal today's webinar is more to focus, give you a broad overview of what is happening in biopharmaceutics. It's a each part in each aspect of biopharmaceutics itself is a separate webinar. But the goal here today is to really, given the context of the international webinar on drug discovery and development, I understand your week-long session going on. It's a nice apt topic for that topic. So my goal is to really give a broad overview of things, and then obviously given the diverse audience that we have today, I've tried to put at a level that it's undergraduate as well as postgraduate as well as somebody who's a little bit seasoned are able to follow the presentation. So typically when most people in the United States, when you go and give a talk, international talk, most people sometimes ask, and even sometimes in the United States, where the heck is South Dakota? So I usually start off with this slide. Most people are familiar with the East Coast, West Coast, California, New York, but South Dakota is in the middle, if you can see right in that, almost closer to Canada. So South Dakota is, I would say, fourth largest state in terms of area, but in terms of population, it's only the whole state's population is close to 1 million. It's not a lot compared to other states. So once you cross out North Dakota, basically you get into the Canada border. So South Dakota, the United States, is famous for mainly, most people come to South Dakota mainly for tourism. As you can see, this is the Mount Rushmore. So it has the 4 presidents, Abraham Lincoln, Theodore Roosevelt, the Thomas Jefferson, and Theodore Roosevelt. So there's 4 famous presidents of the United States. You see in carved in the mountain. So this is what this is famous for. This is in the western side of the state. So the university is located on the eastern side of the state. In fact, when you go from east to west, there is a change in time zone. So we are in central time zone, and the western South Dakota, where you see this mountain, actually is in mountain time, which means it's 1 hour behind us. So within the state, you find 2 different time zones as you pass from one side of the state to the other side. So South Dakota State University is actually, I would say, more than 125 years old. In fact, it is pharmacy school was one of the first schools that was started as a part of the agriculture college, and then it eventually spun into a separate college in the campus. So South Dakota State University is the largest state university in the state. We have 5 universities, and we have roughly 10,000 students for all programs. We have engineering, pharmacy, nursing, arts and science. Agriculture is a big thing. So you see a aerial view of the campus. So Brookings University town of population of 20,000, excluding the students, because we do get pretty cold. As you can see, in winter, we get quite a bit of snow. And sometimes it can go up to below 0, especially in January, February. And then, of course, in the United States, universities are usually, people relate to sports. University sports is very big in the United States. So South Dakota State University is very popular for football. Football here, they talk about more American football, baseball, and of course in basketball. And we also have a event which usually comes in October, which is called as Hobo Day. Basically, it's the whole town is celebrating the team that is usually have a home game, a football game. So it's a big event in the whole city. So all the people in the city would participate in a parade. So that's what you see on the bottom. So the College of Pharmacy is, I would say, is again, so one of the oldest colleges. I mean, not the oldest, but one of the oldest colleges. It is the PharmD. We have a very strong PharmD program. We have it's also allied health professionals. We also have a Master Public Health, which is an online program. We have a bachelor's in medical science, laboratory science, and then also a bachelor's in pharmaceutical sciences. Of course, then we also have a PhD in pharmaceutical sciences. We are in the process of starting a master's in pharmaceutical sciences, which will start sometime in next year. So we have 3 departments: Department of Pharmaceutical Sciences, Department of Pharmacy Practice, and Department of Population Allied Health. So the pharmaceutical science, one of the main focus, I would say, is cancer research and all aspects of drug discovery and development. We also have people working on neuropharmacology. Of course, drug delivery is a big focus in the department. And we also have a faculty working on eye research and looking at corneal injury and drug delivery to the eye, drug development for the eye. And then also recently, 2 or 3 years ago, we added another discipline, genomics. We have a faculty working on looking at genetic basis of diseases and then trying to develop better therapies. So we are lately, I would say, a smaller department, but very highly integrated and engaged department. Our PhD is an integrated program in pharmaceutical sciences, which means we give the breadth, but also the depth in a particular area of research, and especially like pharmaceutics, pharmacology, medchem. But the goal is to give a broader training in pharmaceutical sciences, but then with the in-depth on a particular area of research. So it's a well-rounded program. Now, as far as the PharmD program is concerned, we are number 1 in the nation, in the United States. We have 140 colleges of pharmacy, number 1 in terms of the NAPLEX pass rates. So NAPLEX pass rate is a national board exam pharmacy, which is required to practice pharmacy in the United States. So our pass rate, because of the quality of students that we get and quality of faculty we have been 98 to 100% pass rate for the last 20 years. So consistently we've been in the top 5. In fact, this year we are the top pharmacy school in the country. The next closest University of Washington, which is a much bigger university than us. So definitely we pride in ourselves into the quality of education we provide to our students. Our PhD is 10 years old, but we have a pretty robust alumni. Around 30 students have graduated working in industry, FDA, academia, both here in the United States as well as outside the United States. So as far as Dr. Namari talked about, mentioned the master's and PhD program, but I thought I'll talk about the beginning rather than at the end. So, so in any school of pharmacy, especially students in the bachelor's in pharmacy or master's, if you're looking to join a PhD or a master's program, majority of the schools will require TOEFL is required. That means test of English as a foreign language. It's absolutely a must, pretty much across the board, no matter which program you go to, for all international students, where English is not the first language. There are certain countries which are exempted, but India is not one of the countries. So although we speak pretty good English, but still TOEFL is required. So at SDSU, our the College of Pharmacy requirement is 79 is the minimum score that is required for admission to the program. As far as GRE, graduate record examination, it varies from school to school, and the score that is required also varies. Some schools don't require GRE, but although all schools require TOEFL. So GRE is basically a examination tests your analytical ability, your ability to critical thinking, and more aptitude for research. So it is general, it's not specific to pharmacy, but there are also subject GREs, but most schools accept general GRE, which which covers all aspects of the analytical and writing skill, as well as the critical thinking skills. So at SDSU, our minimum requirement is 140. So we also look at the ranking, percentile ranking, and where the student stands. So, but in terms of requirements, some schools most of the schools require GRE, but the scores can vary from school to school. So for students who are looking to come to the United States, so definitely if you're interested, you should look at TOEFL. And also it's a good idea to research the school and try to find your interest. Instead of sending a generic letter, make sure you look at the faculty's research interest and express your interest, why you're interested in a particular program or a particular faculty. Obviously, it's very competitive. So in our program, we only take 20 students in PhD, and all of them get stipend. They get an annual stipend. We don't take any students without stipend, so which means it's highly competitive. Typically, we admit maybe 1 or 2 every year, depending on the availability of funds. So our maximum strength is 20 students. But we're also starting the masters. At the masters, we are planning to start as self-funded. That means the students pay full tuition. Although they pay full tuition, but when they come to the campus, they can find on-campus jobs to support themselves. So again, it varies from school to school, but most of the programs, PhD programs, usually they're supported by stipend, but also they're competitive. So it's always a good idea to research the schools. And also if you have masters and publications, even always helps. But some schools even admit directly from bachelor's as well if they have a good academic credentials. So that's just about general introduction. So just for the audience, especially the student audience, hopefully that's helpful. So now I'll switch gears and talk about the role of biopharmaceutics in drug discovery and development. So before I start, I wanted to you to kind of give you a contextual perspective of how the drugs were developed early on, and some of you may be familiar with this slide. This is the Banting and Best, the discovery of insulin in 1920s. Now, if you think about how insulin was discovered, it was more of a trial error. They knew something in the pancreas was reducing blood glucose. They didn't know what it is. It used to be called as iletin because of where it's secreted from islet of Langerhans. And this is a dog that was depancreatized dog. That means the pancreas is removed, and so it developed diabetes, and they were able to treat the first dog. So there's an interesting story about insulin, how they developed. It was more of a luck rather than by choice. Carl Best was a fourth year medical student. So he was supposed to go on a vacation, but he decided to work with Frederick Banting, and he had only one opening. And he and his friend flipped a coin, and Carl Best won the toss and went on to work on Banting, and rest is history. Both went on to win the Nobel Prize in 1922 for the discovery of insulin. During the same time in Europe, if you look at discovery of penicillin, that revolutionized the whole antibiotic world. That's the first antibiotic that's ever developed. And then again, as you know, it's an accidental discovery. There was a mold growth that was seen in a petri dish, and it was thrown to the trash can. And then he found something interesting, Alexander Fleming, and that led to a whole set of antibiotics. The reason I put these two drugs, both the drugs revolutionized medicine. Insulin is the first protein that was developed, and now we have a lot of monoclonal antibodies and antibody drug conjugates. On the other hand, you have antibiotics. Again, it's a breakthrough. It opened up a new era of research, but both of them are more of an accident or not by a rational drug design. But definitely, drug discovery changed over the times, and it has become a lot more rational, systematic, rather than just by chance. In fact, if you look at biology, of course, the information flows from DNA, RNA to protein. But you look at the discovery, it was the reverse order. So protein in the insulin, which was discovered in the 1920s, it became a therapeutic agent. DNA of Watson and Crick in 1960s, double helix, and then the Craig Venter and Francis Collins, who is the NIH director currently, the unveiling of the human genome map in 2000. So it evolved into what is called the biotech era. But it all started way back in 1920, and then the DNA discovery as well as the genomic map has opened up a huge amount of data, but also a number of druggable targets, which has made a drug discovery a lot more a lot more rational in terms of finding the right target for the right disease, and also making the right compound to treat the disease. So drug discovery has evolved over a period of time. If you look at the early 50s, it was primarily driven by pharmacology and synthetic organic chemistry. There was no discipline called biochemistry till 1960s. And then the 60s, 70s are mainly driven by a lot of physical chemistry and organic chemistry developments. And computational chemistry came into the field in around 80s. And combinatorial chemistry really opened up, especially in the industry side of things, to screen thousands of compounds in a given day, also synthesize, but also screen using high throughput screening. But as we went to more from a discipline specific, especially with the unveiling of the human genome map in 2000, drug discovery has become pretty much driven by biology, and still today it is very much a biology driven, because we know a lot more about targets. So we know we know a lot more about the biology to rationally design drugs, as opposed to just designing molecules and then retrofitting to the target. So of course, drug discovery also has become costly and expensive, and a lot more tedious in terms of time consuming. So of course, it starts with the target validation, but also lead development, lead optimization, and of course, a big chunk of time is spent in clinical trials and getting the drug approved. So if you look at the academic versus industry research, again, this is the context of the United States. Some of them may apply to India, but some of them may not. So if you look at the academic research, I assume most of the audience today is from academia, so they may appreciate the fact that academic research has a pretty significant role in drug discovery development. So what you see is the triangle here on the pink and the green is academia is very strong in the basic research, whereas the industry is more strong on the resources to conduct high throughput screening. Of course, we are very strong on the foundational basic research, which eventually leads to a product in the long term. So a lot of the institutes are getting in translational research. Of course, very, very few institutions can, academic institutions can do clinical research. In the United States, some universities which have medical centers associated with the university, they are able to do some clinical research, but again, they're very limited. On the other hand, if you look at the industry, they very much apply. Basic research is very limited. Of course, they are a lot, I mean, they can translate from the academia to industry, but they're very big on clinical research. They have the resources, money, and people to do the large scale clinical trials, which is impossible in academia. So definitely it's a good combination. So on the right hand side, what you see is the different aspects where the blue indicator indicates where the academia is better than industry, and there is the orange or the yellowish is more where the academia is better than the industry. So if you look at that, for example, medicinal chemistry, of course, academia is very strong on the medicinal chemistry and assay. The industry is strong in medicinal chemistry with the high throughput screening and combinatorial chemistry. Same with the screening of the compounds. And for stability, job stability, as I know a lot of you look at industry as academia. It's kind of split, but I would say academia is a lot more stable compared to the industry in terms of job. In terms of aligning the needs of the society, of course, academia is a lot more open compared to industry, which is a lot more based on intellectual property. A lot of things are not discussed in public domain. In terms of innovation, a lot of the ideas generate from academia and then goes to industry. So the actual idea, a lot of the drugs that currently have in the market, they all started from the academic research and eventually got into the market. The industry took it from there and developed it further. Of course, disease expertise, it depends on the type of institution. It's a lot of medical schools in the United States are very strong disease expertise. They focus on disease, whereas industries, depending on the company, they might focus on certain disease, but a lot of the basic foundational disease aspects comes from academia. So definitely academia has a lot more offered drug discovery development, and in some cases more, and sometimes equally, if not more, than the industry. So some of you are familiar with the slide in terms of the drug discovery development. This is a slide from Pfizer. It starts from idea, then you do a lot of early stage exploratory research, and you make a lot of compounds, and then screen the high throughput screening, and a lot of them get screened in the preclinical research, and then only a few compounds advance to clinical trials, and it's a tedious, long process before it gets to the market, before it gets approved by the FDA and gets in the market. So, of course, it's a very risky and expensive business. If you start 10,000 compounds, actually, you may end up with one approved drug. So a lot of the attrition happens as it goes from drug discovery to preclinical to clinical trials. Of course, the time scale is also equally important. It takes 12 to 15 years. Now, although the number is 1.2 billion, the recent estimates around close to $2 billion. So $2 billion, and the chance of success is 1 in 10,000. So basically a needle in haystack. So it's a very expensive, tedious process. So as you think about this, so why don't all drugs make it to the market? So what are the reasons? Obviously, industry is asking the questions, and they've been asking for a while. They would definitely want to succeed with putting less resources, but less expensive, but at the same time, reduce the risk. So if they want to fail, they want to fail early. They don't want to know it till it goes to clinical trials. So if you look at the reasons, again, this is a lot of research, Pfizer being the largest R&D in the world, a lot of research has come from them. If you look at the reasons for why the 9,909 compound don't make it to the market, they split the reasons. So if you look at this chart, pie chart, around 40% of the drugs don't make it to the market because of poor pharmacokinetics. I would say poor biopharmaceutics and pharmacokinetic properties. Now, 30% of drug is due to lack of efficacy. Of course, they work very well in animals, but the moment taken to clinical trials, it doesn't work very well. Then you have other reasons, well, toxicity and so on and so forth. But this is the important thing. 40% of drugs don't make it to the market because they have poor delivery properties, which means poor biopharmaceutical and pharmacokinetic properties. So this tells you something about the importance of talking about biopharmaceutical and pharmacokinetic early on. And the industry has recognized this. Earlier, they used to make a compound and then test the delivery property later on. Now it is shifted. Now the focus is what they call the physicochemical profiling. They do it early on to screen the best compound. Not only the activity is important, but also equally important is the fact that you're able to deliver the drug at the right target, the right dose, the right time. So it's very important aspect that need to be taken into account early on, not an afterthought. So I want to give an overview of, and especially this may be some, a very basic for some of the student. I mean, for the student's sake, I put the slide. So if you look at where do you start from, and as you think about pharmaceutics in whole, I call what I call the 4Ds. In fact, pharmaceutics, you really have to integrate all the 4Ds: the disease, the drug, the destination, which means where you want to deliver, and of course the delivery properties. So you need to understand the chemical, physical properties of the drug. Of course, you need to understand the disease. It's not a one size fits all. You need to understand the pathophysiology, at least some understanding of it. And then you need to know where you're going to deliver, systemic or local. Of course, and delivery, the biopharmaceutics, pharmacokinetics, and other delivery aspects come into play a big role. So you don't develop a delivery system and then think about what drug to retrofit in. Rather, you wanted to develop the right delivery system for the right drug and the right disease. So that's very important. So the 4D approach is an important aspect to understand the drug discovery development. So I often say that pharmaceutics people are jack of all trades, master of none, which means you have to know a little bit of everything in order to develop a deliverable drug or a druggable compound. So if you look at the how the drugs are developed, so basically, of course, they hit on different aspects of the 4Ds. So one is called the market pull approach. That means there's a clinical need where there's no drug as of now. So that is a motivation for developing a new drug as well as a delivery system. Sometimes it is the technology pull. In other words, already drugs on the market, but the current route of administration is not optimal. So then you go back and look at the drug properties, modify the drug or modify the delivery system to make it a better drug in the market. So I often say the pharmaceutics is the abilities to address the ability. So you talk about solubility, permeability, stability, deliverability, manufacturability, and bioavailability. Of course, the last one is very, very important. You should be able to deliver the right amount of the drug at the right time in the right target site in the body. So of course, pharmaceutics has three aspects. Physical pharmaceutics, very, very important from a pre-formulation or physical profiling. Then you have biopharmaceutics, which we'll talk about today. And then of course, pharmacokinetics, which is it's related to the biopharmaceutics, but primarily the ADME, absorption, distribution, metabolism, and excretion. All the three need to be considered in order to develop an efficacious and a safe formulation or delivery system. So when you think about biopharmaceutics pharmacokinetics, the simplest way to think about it is a Ladber system, which is basically outlines all the steps the drug has to go through before it can deliver to the site. So liberation, the drug need released, so it didn't be absorbed the right amount at the right rate, distributed in the body, metabolized, eliminated, and then of course the pharmacology of pharmacodynamics. So the release and the absorption, that is the aspect that we are most interested in biopharmaceutics. Of course, the kinetics of absorption, distribution, metabolism, elimination is pharmacokinetics, and pharmacodynamics, of course, is the drug's pharmacological action. So and most of you must have heard this. So pharmacokinetics is what the body does to the drug. Pharmacodynamics of pharmacology is what the drug does to the body. So I call biopharmaceutics what you do as a scientist, as a pharmaceutical scientist. What do you do to the drug in order to make it a better drug or deliver it to the body at the right time at the right dose? So you have three aspects: biopharmaceutics, pharmacokinetics, pharmacodynamics. All the three need to be considered in tandem. Now, if you look at each of the steps, and I've just outlined some of the physical properties, invariably, a lot of them repeat. As you can see, solubility is a big part of the dissolution. Of course, it comes in other aspects as well: distribution, log-P, the lipophilicity of the drug, pKa, molecular weight, chemical structure, in all the steps. So this is very critical. In other words, the physical property is what is the most critical parameter in optimizing a drug. In other words, for a drug to be druggable or a drug to convert to medicine, you need to really consider the physical properties, because it influences pretty much every step of the way, all the way from the release of the drug to the final response. So biopharmaceutics is study of the influence of physical properties and drug absorption. So primarily the drug absorption, and you'll see in a minute why I talk about drug absorption so much. So if you look at the different routes of administration, except intravenous, pretty much all the routes, the drug need to cross the membrane in order to get to the blood. Because once you get the blood, it gets distributed, get excreted in different body fluids. But of course, the liver and kidney plays a pretty significant role in excretion of the drug, elimination, metabolism, as well as the excretion, right? So except intravenous, which can directly get the drug into the blood, pretty much the drug has to cross several biological membranes before it reaches systemic circulation. So it's very critical to understand the barriers. So why did I talk about absorption so much? So if we look at the different routes of administration, of course, the oral route is the most convenient and the most popular route of administration because it's easy to administer drugs. A majority of the products, around 40% of drugs in the market, are oral products. Of course, you have other routes as well. And as you see, most of the routes, or all of the routes, actually require the drug to be absorbed through a biological membrane before it reaches systemic circulation. Not only that, even beyond that, getting distributed also requires it to be distributed. It has to cross membranes to distribute different organs of the body. So majority of the drugs are delivered through oral route. So that is why the major focus is on the oral drug absorption. So if you look at a typical plasma time profile, so most are familiar with this curve, I'm assuming. So you have the Tmax, time to reach the peak concentration, Cmax is the maximum concentration, elimination rate constant, and you have the absorption rate constant. Now, then you have therapeutic window. Now, what you can control, what I mean to say, we in the sense of pharmaceutical scientists can control, is the left-hand side of the curve, right? Once the drug is absorbed, the body will take care of it. But whatever you do, formulation, delivery system, route administration, everything is the left-hand side of the curve by altering the physical properties of the drug as well as the formulation. So biopharmaceutics is nothing but basically studying the left-hand side of the curve and optimize the drug. So which is primarily focused on the bioavailability. Of course, the plasma time profile is pretty much looks similar. Of course, the rate and extent may vary depending on the route of administration, except intravenous where you don't absorption. So bioavailability is the rate and extent of drug available from a dosage from the blood. Of course, how fast and how much, the rate as well as the extent is important as you think about biopharmaceutics as well as bioavailability. So what are the barriers? So most of my discussion will focus on the oral because of the reason as I just mentioned. So the drug need to, irrespective of what the dosage form you give, the drug need to disintegrate, dissolve, and need to be soluble in the biological fluid. The drug need to be in solution. Of course, how long it stays in the stomach or the intestine, it depends on the GI transit time, which means food influences. It's typically 4 to 6 hours in the small intestine, 1 to 2 hours in the stomach. So not only the drug need to dissolve within the time period it is in the body for it to be absorbed. Only the drug in solution can be absorbed, except some particles which can be absorbed by a process called endocytosis. But predominantly, all drugs need to go into solution before it can be absorbed. Of course, you need to be stable in the biological fluid in the different varying pH of the stomach and the intestine before it can get absorbed, so it shouldn't get degraded. Then you have the mucous layer. It has to cross the mucous layer before reaching the membrane. And again, it has to have the right lipophilicity or log P to cross partition to the biological membrane. And within the membrane, what we call the enterocytes, the drug may be metabolized by enzymes. A lot of enzymes and a lot of transporters. We'll talk detail as we get through the presentation. But you need to cross this barrier. And even before getting systemic circulation, it goes to the liver to the portal vein. So some of the drug may be lost by what is called a first pass elimination in the liver. So the remaining is what you get in systemic circulation. So there are several barriers. You have drug need to be soluble, drug need to be stable, drug need to be permeable, drug need to be metabolically stable, and both in the intestine as well as in the liver before reach systemic circulation. So most are familiar with the different phases of drug metabolism. Phase 1 is mainly oxidation and reduction, those kind of reactions to defunctionalize the drug. And phase 2 is mostly conjugation reactions. But the advance in biology, what we now understand is the fact that not only the phase 1 and phase 2, we also talk about phase 0, even before getting into the intestine or into the liver, which is called as basically the uptake transporters. Transporters play a very important role. Not only the uptake, but also efflux. Something kick the drug from the membrane into back to the intestine, which is called as efflux transporters. So this aspect is in drug disposition is called as phase 0, even before the drug metabolism. Enterocyte is your intestinal cells. Of course, the efflux transporters can be expressed either side. The drug can be effluxed from the apical side into the intestine or on the blood side into the blood in portal vein, which is the vein that supplies the blood to the liver. Within the liver, then you have again uptake transporters and efflux transporters. It has to cross the barrier. So this is called as we call the phase 3 metabolism. Phase 3 is basically the aspect that is primarily involved into the efflux transporters on the other side of the enterocytes or in the intestine into the systemic circulation. So phase 1 and 2, most are familiar with in terms of the drug metabolism. Phase 0 is the efflux or the uptake transport on the apical side. Phase 3, the efflux transport is on the other side, which is the blood side of things. Of course, in the liver, we only mainly talk about liver metabolism, but also equally important is the excretion of the drug into the bile, because liver is where the bile is produced. Some drug can be excreted in the bile, and then bile goes into the intestine, empties into the intestine to the drug may be excreted in the feces or can be reabsorbed depending on how it's broken by the bacterial enzymes in the colon. So just to give you an idea of how many barriers the drug need to go through, so I give an example of cyclosporin here. So cyclosporin is a drug which is it's an immunosuppressant drug. So if you look at cyclosporin, the extent of drug absorbed is 86%. If you have 100 million of the drug, 86 million of the drug is absorbed. 14% of the drug is excreted as it is in the feces or degraded in the intestine. Among the 86%, around 51% is actually degraded in the intestinal walls, even before systemic circulation. And the remaining, there are 88% of the drug also undergoes first pass. So eventually what you get is bioavailable drug is only 27%. Some of it is also effluxed by efflux transport in the intestine. So you can see that there are several barriers the drug need to, it's not automatic, the drug that's everything that you give doesn't necessarily mean that it's going to be systemically available. So there is definitely a difference between absorbed dose and available dose. So absorbed dose is what is absorbed to the intestine, but not all the absorbed dose go to systemic circulation because of the metabolism that's happening in the gut wall or the intestine as well as in the liver. So it's very important difference between absorbed doses and the available dose. So if some can please mute the microphone. So as you can think about the different aspects of the gastrointestinal tract, of course, the varying pH, different transporters. Certain transporters only expressed in the intestine, some are expressed in the small intestine, large intestine. The different varying fluids, of course. Then the length of the GI tract varies from different segments of the gastrointestinal tract. So which also means that drug stays in different for different lengths of time in different parts of the GI tract. So that's very important to understand the physiology of it. Now, some of you, especially the master's student, may be familiar with the fixed law of diffusion, which is a fundamental law that governs the drug permeation through membranes, biological membranes, as well as synthetic membranes. So this basically sums up all the factors, physicochemical as well as physiological factors that influence drug absorption. So basically, it says rate of absorption and rate of transport is influenced by the diffusion coefficient of the drug, which is function of molecular weight, as well as the membrane, how thick the membrane, how well the drug is able to diffuse, the surface area that's available, of course, the drug as well as the biological membrane. For example, small intestine has the largest surface area because of the microvilli or the villi, which is a finger-like projections in the small intestine, which you don't have in the stomach. So that has the largest surface area. That's why most of the drugs are absorbed in the small intestine as opposed to the stomach or the large intestine. Partition coefficient is important for the permeability of the compound. The concentration of the drug in the GI tract is important, which is governed by solubility. Of course, the denominator, which is the thickness, H is the thickness of the membrane. So that again is also influenced by the where the drug is absorbed, where the small intestine was the large intestine was the stomach. So pretty much all of the factors, the molecular weight, the physiological aspects of the membrane, the absorption mechanism, the dosage form, of course, the dissolution of the drug and the content of the drug in the GI tract fluid. So both the rate as well as extent is influenced by one or more of the parameters. So as a drug discovery development scientist, you're trying to alter one or more of the parameters in the numerator. Of course, you cannot alter the thickness of the membrane physically, but you can actually localize the drug to the stomach or the intestine based on the available absorption mechanisms. But most of the focus is on how to alter one or more of the factors in the numerator to make a compound druggable. So these are different mechanisms. Most of the drugs are passively absorbed across the cells or between the cells, and some are taken up by transporters with energy, which is called active transport. Without energy is more of facilitated transporters. For example, like some vitamin B12 as well as some antibiotics like cephalosporins. And if there is a drug is not completely dissolved, they're taken up by a process called endocytosis. It's called cell drinking. Basically takes up the particles, and it's a very minor transport mechanism, but this can also function in the intestine. In addition to influx, there means absorption transporters. There is also things which can kick the drug out of from the membrane back to the intestine. They call efflux transporters, like example, P-glycoprotein. So you need to also take this into consideration. In fact, this plays a very critical role in low bioavailability of compounds. A lot of compound hydrophobic or lipophilic compounds. The drug gets absorbed in the membrane, but before it reaches the other side of the membrane, the transmembrane protein, P-glycoprotein, by an active process using ATP, it kicks the drug out of from the membrane back into the intestine. And the other interesting thing is there's also CYP3A4, which is the cytochrome P50 enzyme, which is present right beneath the PGP. It is a protective mechanism the body has developed. So even if the drug escapes the PGP, the drug metabolizing enzyme will metabolize the drug. So in other words, it's a cycle that goes on. So the drug is kicked out, absorbed, again kicked out, and most of it is either kicked out or metabolized by the drug before reaching the systemic circulation. I see some. Okay, thank you for that. So it looks like somebody has a host option open. So Dr. Ramani, are we doing good on the time?
Please go ahead. It's going good.
Okay. So then just to give you a quick overview of the different transporters. So you have different types of facility transporters. One is called uniporter. For example, like the OCT is the organic cationic transporters. A lot of things we know, most of them are designed for absorption nutrients, but we are designing drugs to match that structural features in order to be taken up by those transporters for better absorption. Uniporter is only one direction, unidirectional. Only one molecule is transported, or one type of only one class of molecules are transported. Then you have what called symporters. That means there are two things that transport at the same time. So it's also called ion coupled transporters. For example, sodium glucose. So the sodium gradient also transports glucose along with it. So some of the peptide transporters, for example, the peptide-1 is one of the peptide transporters, which is involved in drug transport, is a symporter. That means two compounds are transported the same direction. Then you have antiporter. One is towards inside the cell, the other one's outside the cell. For example, sodium potassium pump is an example where one is inwards, the other one is outwards. So there are all set of transporters that are being identified, which are involved in drug transport. Of course, they're all designed for nutrient transport, but now we have designed drugs to be have same structural features so that can increase the drug absorption, especially for drugs which are poorly absorbed. In fact, it's been studied and also now established. Although we initially considered most of the drugs are absorbed passively, that means they don't involve transporters. It's believed now that pretty much every drug is absorbed by transporter. The fact that we just are not identify the transporter one way or other. So, but of course, still the research is still ongoing as we study more about biology. There are a lot more transporters will be discovered. In fact, we have 300 or 400 families of transporters we know as of now, both either involving facility transport without energy or involving ATP. So just to give an example of the ATP transporter, which is also called ABC transporters, that means they involve ATP, ATP binding cassettes, they're called. P-glycoprotein is not an absorption transporter, but efflux transporter. This is the one I was talking about. So drug gets absorbed, kicked out, again, keeps on going on. It has been known that drugs or substrates of PGP are all substrates of CYP3A4, which is the enzyme. So which means they work in tandem. If the drug escapes anything from PGP, they get metabolized. So most of it's lost or metabolized even before reaching systemic circulation. So here, just to give an idea, so there are several drugs. In fact, not only the drugs, but also drug and other food substances can interact. They are substrates for PGP. Now, these are some of the drugs which are in the market, which are either purely substrates. Some can function both, like rifampicin substrate as well as inducer. It can induce the PGP. And some are inhibitors. So for example, piperine, which is a natural compound, of course. And you have a lot of the drugs in the market, like quinidine, for example. So you see there are a lot of interface. So there's a lot of overlap. Inhibitors, some substrates can also be inhibitors, some substrates can also be inducers. So that means a lot of drug-drug interaction, which can not only involve drug-drug interaction, mostly talked in the context of pharmacology, but a lot of ADME, which means they can interfere with the absorption, distribution, elimination, as well as efflux transporters. So this is just a snapshot of some of the different transporters that are involved. As I said, there are this family number of families are increasing depending that proteins which is a sequence proteins amino acid sequence in the protein. SLC are solute carry transporters, which most of them don't involve energy, or they're ion coupled transporters. Then you have the ATP binding, which involves pretty much ATP in the transport. Mainly they are reflex transporters. So there's a whole set of transporters. This is evolving. As we speak, there is more and more transporters being identified, which are a lot of the drug substrate for different transporters, as you can see in this table. Now, when you talk about transporters, it's very important to understand not only the type of transporters where they express. Are they on the apical side or on the blood side? For example, this is the intestine. So as you can see from the arrow, some are unidirectional, some are bidirectional. For example, the cationic transporter, monocarbose transporters. MRPs are efflux transporters, multi-drug resistance protein transporters. LAT is a lactic acid transporter, both directional, bidirectional. Some are not only efflux transport expressed not only the apical side, but also on the basolateral side. So it's very important to understand what transporter, which side of the membrane, because that dictates how much of drugs gets absorbed or kicked out of the membrane into the lumen or into the blood. The same thing with the blood-brain barrier. You have expressed both side on the luminal side as well as on the basolateral side of the blood-brain barrier. And now in hepatocytes in the liver, now as you can see, again there are a lot of transporters. Now it's not automatic that the drug that gets to the blood gets into the liver. Majority of them gets to what is called transporters. It's not, drugs cannot get through passively. They need to take in by transporters. That means the expression of transporters dictate how much the drug gets metabolized. Not only that, some of the drugs also get into the bile based on transporters. BSEP. BSEP is the bile salt export protein, which takes the drug from the hepatocytes into the bile. The bile then goes to the intestine and gets excreted in the feces. So transporters are not only involved in absorption, but very much involved in metabolism as well. Same thing is true with excretion in the kidney. This is a big area of research. In fact, FDA has asked a lot of information about kidney toxicity, liver toxicity. People believe the transport is involved in drug-drug interaction as well as drug toxicity. So again, a lot of transporters, both efflux and absorptive transporters, into the renal cells, but also into the urine, the lumen of the kidney. So as you can see, transporters are ubiquitous and different organs of the body. I just gave you only 4 organs just to give an idea. Of course, both the eliminating organs as well as the absorption organs, there is significant transport expression which dictates how much of drug gets absorbed, metabolized, and distributed. This is just to give us a snapshot of the different transport of the regulatory agencies usually ask for, FDA as well as the European Medical Agency. I've just given an example. They talked about some of the substrates that you have to use to characterize those transporters. And here again, you see the chart of the efflux transport, uptake transporters, and depending on the extent of metabolism or excretion with the kidney, different transport need to be characterized for specific drugs as you develop the new drugs, both the FDA as well as the EMA, European Medicine Agency. So as you can see that it is not only just academic exercise, but very much involved in getting regulatory approval from for new drugs. This is again a decision tree about for studying substrates in a use blood-brain barrier, usually cell culture models. Rat brain endothelial cells. Cacoto cells very, very popular and very commonly used for intestinal cells. Cacoto is colon carcinoma cells used for studying membrane transport. And then you have hepatocytes used for liver uptake, and same with the proximal tubule cells especially for rat cells. Then we're talking about substrates. If you're looking at uptake versus efflux transporters, depending on the permeability of the compound, different methods are used. ATP assay to understand the ATP-driven uptake. Bidirectional transport assays or vesicular transport. I'll not go into details of the methods, but just to give an idea. Of course, in some you have to also do inhibitor studies, both for efflux and uptake, to characterize the transporters that involve transport of drugs. So very comprehensive decision tree depending on a substrate or depending on efflux or uptake, as well as whether you're looking at inhibitor to further characterize the drug transporter uptake or uptake of drug by the transporters. So this is kind of a snapshot of some of them can be predicted in silico as you look at permeability as a transporter characterization. Then you do in vitro studies in a cell culture model, both uptake as well as completely grown monolayer cells. And then of course you can do an animal studies, in situ perfusion studies in intestines, as well as in vivo studies in animals and pharmacokinetics, both to understand the toxicity as well as the drug interactions. But then finally, of course, in human clinical trials as well. So you start from simple prediction, in silico prediction, to all the way up to the human studies. I'm just going to show you some examples of methods that are used. So the most commonly used cells for intestine is the Kakoto cells. This is a transmission electron microscope of the Kakoto cells. They do have similar architecture, the actual intestines, like the finger-like projections, but not as extensive as what you see in the intestines, but pretty close. So the nice thing about Kakoto cells, you can grow them, you can express them by, express certain genes, whatever transport is interested in. So you can basically study by knockout or expressing the particular protein, which transporter proteins are involved in transporting the drug. So it's a very easy to study in high throughput screening. You can see thousands of compound, 96 cell plate. So it's very, very widely used. Of course, there is some limitations, which I'll talk about later in the presentation. One, it does not express certain enzymes, so you have to express them in order to study the metabolic profile of a drug in the intestines. You can also use a membrane level, like aborted rat intestines. Basically, you revert the intestine, you take the intestine out. If it's passive, you can go both bidirectional. But if if the membrane is inside out, the transporter expresses on only one side. So if it is specifically taken with transporters, then you only see when the transporters. So the drug is kept on the outside. The drug is kept on the inside of the intestinal bag and then outside the buffer. So if it's passive, you can go either side. But if it's an active transport or facility transport, if the transport is outside, the drug cannot transport it. So it basically tells you whether it is actually transporter mediated or passively transported. Then you can also look at a live rat and anesthetic rat, and then you can use a perfusion pump to understand the transport or absorption of a particular segment of the intestine to see where the drug is absorbed. You can do the same studies in the humans as well. It's a lot more complicated. You do a balloon catheter to just block part of the intestine and then infuse the drug to study the drug absorption and take the blood samples to study the drug absorption when the drug is infused in a particular segment of the intestine. As far as the correlation goes, so how well does the Kakoto permeability compare with the actual in vivo studies of fraction absorbed? So what you see here is, this is the one I was talking about, some of the limitations of Kakoto cells. So the drugs which are poor, so this is basically the squares represent all the data that generate for drugs from the Kakoto cells. And this is the permeability of the actual intestinal, human intestinal, doing an in vitro study, taking the human intestine out and studying in vitro. And this is the correlation between the human intestinal permeability, the Kakoto versus the actual in vivo absorption in the humans. So what you see is compounds are very well absorbed or very well permeated. They're pretty close. That means Kakoto cell predicts very well the membrane permeability. But for poorly permeable drugs, they're wide apart, which means they are not good predictor for drugs that are low permeability. The reason being, Kakoto cell is pretty much a flat surface, as opposed to the actual intestine, which has a lot of finger-like projections. So in other words, the poorly permeable, if the drug is shown to be poorly permeable in Kakoto, does not necessarily mean poorly absorbed in the actual in vivo, because the drug can go between the villi and get absorbed. So the actual in vivo absorption may be much, much higher than what you see in Kakoto cells. But if the drug is highly permeable, majority of the drugs, they show very good correlation in the in vivo because they're very well absorbed, they're rapidly absorbed. So it doesn't matter whether Kakoto or the actual in vivo, they're rapidly absorbed, as opposed to poorly permeable drugs, which take a lot more time and gets into between the villi to get absorbed. So in other words, in a nutshell, Kakoto cells are a good predictor for highly permeable drugs, but not so much drugs that are poorly permeable. In other words, when you see something's poorly permeable, doesn't necessarily mean that it's not going to be a good druggable compound. So you need to be a little bit careful to interpret the results, especially if the drugs are poorly permeable, because it may be very well absorbed in the intestine because you have a lot more villi in the intestine compared to the Kakoto cells. How about the rat permeability? So this is rat versus the human intestinal permeability. They have very good correlation, especially passively absorbed compounds. Again, the human in vitro intestinal permeability very well correlates with the fraction absorbed in the in vivo. So it has been found that the drug need to have at least 2 to 4 centimeter square permeability in order to have good absorption. So as you can see, if it's 2 to 4, almost 90 to 100% drug is absorbed in vivo. So in vitro tests can very well predict what is going to happen in vivo. Now, there is also some things you need to think about as you think about rat and humans. There is definitely transport expression rat versus the human duodenum. Of course, there is a reasonable correlation about 0.56, but the level of expression is very different. Some are higher, some are lower in the rat compared to humans. Not only that, the transporter, but also enzymes. As you can see, the enzymal expression, there's not much correlation, which means the levels may vary. Not only they vary, but depending on the location of the GI tract, it can vary. For example, in humans, the MD multidrug resistance protein is high in the duodenum compared to the colon, but in the rat, the colon has more than the duodenum. So the location is also variable compared between the rat and humans. And again, CYP3A4 is an enzyme. Again, in the rat, it is very high in duodenum compared to the colon. In humans intestine, it is high in the duodenum and pretty much none in the colon. So very important to understand the differences or the limitations of the method that you're using. But still, overall, it is still the rats can be a good predictor of absorption in the humans. So the question is, when you talk about so many things, so what are the two most important aspects? So of course, all of them are important, but if really it comes down to two things, if you relate all of them, is the drug solubility and permeability the primary determinants of whether the drug will be very well absorbed or shows good bioavailability. Very, very critical. This is what we try to optimize in drug discovery and development. You are directly and indirectly optimizing drug solubility, make it better soluble, or make it better permeable. You're basically lead optimizing or changing the structure or modifying the structure to achieve one or more of these parameters. So ultimately, this is the drug soluble and permeable to the primary determinants of drug absorption. So in a drug discovery development, of course, depending on how the drug progresses from early stage to the late stage, you do the right tool at the right time. You don't do extensive study initially. You do very limited study, kinetic solubility, Kakoto cells. Then you do equilibrium solubility, and then you isolate rat intestine and perfusion study rat. Then pH solubility profiles. So as you advance further, the number of studies advances as well, or you do expands. Of course, in human studies, you do more dissolution studies. So you go from high throughput to the early stage to low throughput in the later stages when number of compounds are limited, or number of, screen the compounds which are more druggable as opposed to the early stage of drug discovery. So from here on, I'm going to give you just some of the tools that are used in drug discovery development, which helps us in guide to select the best compound to take it forward. Of course, one of the things I talked about, you don't want to spend $2 billion to find out if the drug is going to work or not. You want to find early on. Some you can find out just without doing experiments, just to in silico predictions and looking at the structure. Some of the limited experiments, some combination of theoretical plus the experimental data. And of course, ultimately, the human clinical trial gives you the actual data, but you don't want to take 10,000 compounds to human clinical trials. So I'm going to show you just the evolution of how the drug discovery development, the models have evolved over a period of time. I would say last 2 or 3 decades and where we are currently. So I'll end with where we are currently in terms of predicting the drug properties. So some of you may be familiar with this called Lipinski rule of 5. So Christopher Lipinski was a scientist at Pfizer. So he was very curious to understand why so many compounds fail. So what he did was a very smart thing. He looked at all the compounds. Of course, Pfizer has a big database of 10,000 compounds or so. He looked at the data and saw which ones made the market, which one did not. And out of the ones which made the market, he said, okay, what are the common features of the drug that made to the market? Chemical properties. And this is what he found, came with the rule of 5, RO5, that's in chart is called. Basically, what he found are the 4 properties. Drugs with molecular weight should not be more than 500 Dalton to be orally available. Of course, there are some exceptions. He says these are small molecules. Drugs are primarily passively absorbed. At that time, in early 2000, there's not much known about transporters. Log P should not be more than 5. Hydrogen bond donors and acceptors. And donors should not be more than 5 hydrogen bonds. OH is the molecule. Again, the acceptors not more than 10. We look at all of them, 5 or multiples of 5. That's why it's called rule of 5. Now, what he found was, if any compound violates more than 1, in other words, if the compound does not qualify 2 or more of the properties, then it is very unlikely it will be absorbed very well or show bioavailability. So, but if you really think about all the properties, they directly or indirectly all linked to drug solubility and permeability, right? So basically, Lipinski rule of 5 is very widely used. Of course, there are some limitation of the rule of 5. People have found some because we know now some of the transporters involved, not all compounds are absorbed passively. But given that, but still it is a good primary screen to screen compounds early stage looking at the structure. So it's a good start. Of course, it's not complete, but definitely a good start to screen compounds. Then the scientist Kurtler, which is again a scientist from Pfizer, he came up with the model called the maximum absorbed dose to predict. This is a combination of experimental plus some limited experimental data and the theoretical data. So this is called MAD, which is the maximum absorbed dose. That means what is absorbed. So it doesn't predict the metabolism, just absorbed dose. Next step, the process. So he used some theoretical values. For example, volume of intestinal fluid, which is usually normal, it's 250 ml. That's a glass of water that you take with a drug. Normal gastrointestinal residence time, primarily is only considered intestine because that's where most small intestine, most of the drug is absorbed, around 4.5 hours. And this is solubility, you can do it in vitro, and absorption rate constant, you can use in vitro permeation studies in intestines, rat intestine and human intestine. So you have some in vitro data, some physiological data, known data. If you combine those things for a particular drug, of course, this the volume and the TI is usually constant. The other two values, of course, will depend on the drug. So what he predicted was, of course, solubility in only the pH 6 point, which is the intestinal pH. You can predict theoretically the maximum absorbed dose. So it will give you whether it can, based on the pharmacology, whether this dose is reasonable to expect some therapeutic activity. So that's basically what the intention of the model. So what he found was, if you look at drug permeability and solubility, there is drug permeability varies only 50-fold. If you look at all drugs made to the market, but drug solubility varies of 6 very wide flexibility. In other words, there is very limited flexibility of varying the permeability. In order to change the permeability, you have to change the structure. If you change the structure, it increases the molecular weight. So it can be counterproductive. But on the other hand, you can take advantage of increasing solubility because you have a lot more scope to overcome even some of the limitation of permeability. In other words, even the drug is poorly permeable. If you increase the solubility, it increases the constant gradient according to Fick's law and increase absorption. So in other words, even drugs are poorly permeable, you increase the absorption by significantly increasing the solubility. And I'm going to show you some examples in a minute, but I just wanted to make sure you understand this. So permeability, you can increase, but it's only a limitation. But solubility, there's a lot more scope to improve without changing too much the molecular structure of the compound. That's the biggest advantage. So here is an example. So you can do a lot of permutation combinations. So this is a compound, for example, if you two different absorption rate constants. So 0.0 is very, very low absorption, of course. As you can see here, for all of them, of course, this is keeping the absorption rate constant. As you vary the solubility, almost 1,000-fold. As you can see, increase the maximum absorbed dose, 0.2 to 200 milligram. Similarly, if the permeability is better, 10-fold higher, again, you get further advantage. So to illustrate this fact, there's an example of azithromycin, which was developed by Pfizer. So if you look at this is a compound X did not reveal. Of course, in the industry, the things they don't reveal because of patent and other reasons. So azithromycin and another compound. As you can see, this compound definitely has better absorption than azithromycin. But the reason azithromycin is made to the market, this compound did not make it to the market, is because solubility. Look at the solubility; it's very, very low solubility. Whereas azithromycin has 50 milligram per ml, very good solubility, in spite of poorly permeable. Because the max absorbed dose is almost close to 3 grams here, right? So the point I'm trying to convey here is, if you have pretty good solubility or increased solubility, you can even overcome some of the poor permeability issues and make a compound druggable. So this is an example of where you can increase drug solubility without altering the structure to increase the absorption. Again, you can do a lot of permutation combination. You can basically shift the left-hand side or right-hand side of the curve. So if you, let's say you want 10 milligram kilogram as a max absorbed dose, you can put what you want and then figure out how much solubility you need to achieve in order to achieve this absorption max absorbed dose. Of course, you need to know all the other parameters. The only unknown will be the solubility. So you can do all permutation combination to figure out what should be my target solubility or what should be my target permeability. So this equation helps you to predict with the very limited in vitro data. Now, the next advancement is now how to integrate the physiological and physical properties. So, for example, surface area, particle size plays a role, but also the intestinal permeability. Same with solubility, depending on the pH, not only the pH in vitro, but the pH in the biological fluid. That's important. So the next advancement was what is called as the biopharmaceutics classification system. So they combined all of these parameters to say, oh, can we come up with simple parameters to predict the in vivo from in vitro experiments? So they actually considered the intestine as a tube and used some physical parameters to define the solubility and permeability. I'll not go into details of the physical parameters, but they came with 3 numbers, what is called as absorption number, dissolution number, dose number. So absorption number is in simple is defined as how well the drug absorbed within the given residence time. In other words, if the GA residence time is 4 to 6 hours, if the drug is absorbed within 4 to 6 hours, that means it has a good permeability. In other words, the permeability divided by the residence time. It means the drug should be absorbed within the time frame which is in the intestine. That is what it means. You need to have a high absorption number, which means it should be rapidly absorbed within the 4 to 6 hours. Dissolution number is again relating the dissolution of the drug or the compound with respect to residence time in the GA tract. Within the residence time, 4 to 6 hours, the intestine is the drug able to dissolve. So the higher the dissolution number, the better it's absorbed. Higher the absorption number, the faster it is absorbed. Now the last one is important, dose number. We all know the solubility, but here is the dose soluble in the volume of the biological fluid. For example, 250 ml is the biological fluid, intestinal fluid volume. Is the entire dose soluble? So, of course, in other words, the dose should be completely soluble in the given volume of the intestinal fluid, not for it to be show good absorption. So absorption number should be high, dissolution number should be high, dose number should be low. In other words, the drug should be soluble in less amount of the biological fluid. Then it will be a druggable compound. So they use the 3 numbers to come up with the classification, which I'll talk about in a minute. So here's an example of digoxin griseofulvin. So of course, both made in the market. So one is a potent drug, the other one is higher dose. Griseofulvin antifungal digoxin, the cardiovascular drug. Solubility is pretty similar, or pretty close, I would say 0.015, 0.25. But look at the dose number. The volume that's required to solubilize this dose, you required only 20 ml here. You need 33,000 ml. 33,000 ml means 33 liters, which is very high, right? So dose number is very high. So you significantly need to increase the solubility in order to make it. So a lot of the griseofulvin that is available in the market is actually use a technology to solubilize to make sure you are able to solubilize the drug in lesser volume. So that's just to show you just similar solubility but different dose number. So digoxin, of course, more potent. The dose is not only the dose, but the amount of volume that is required to solubilize the drug is equally important. So anyways, going to the BCS, Biopharmaceutics Classification System. So Gordon Amida and University of Michigan, who came up with this classification in 1995, which actually revolutionized the whole biopharmaceutics field. He classified the drugs into 4 classes based on market drugs, not the new drugs as a roadmap for the new drugs, but mainly based on the market drugs. Drugs fall in 4 categories. Class 1 is highly soluble, highly permeable. Perfect class and ideal class. You want most of the compounds to be here. Class 2, permeability is not an issue, solubility is an issue. That means you need increased solubility. Class 3 has good solubility but low permeability. Class 4 is very, very difficult to formulate or make it a druggable compound because it has low solubility and low permeability. So this chart shows the different pharmacopoeias around the world, European, German, and European pharmacopoeia, as well as the Japanese pharmacopoeia. They have 2 different volumes of dissolution. That's why 2 different colors. So as you can see, majority of the compounds on the market fall in class 2 and class 3. There are a few compounds class 3, very, very few compounds class 4. And then, of course, there are some unclassified or classified in multiple classes. So he defined solubility as the highest dose strength that's soluble in a glass of water, varying in pH from 1 to 7.5, which is the entire range of the gastrointestinal tract. They are considered highly soluble. Any compound shows more than 90% bioavailability is considered highly permeable. So of course, a lot of criticism about this classification because it's very stringent because very few compounds fill in this category, but this is of course what they came up with initially. FDA uses this to use mainly for waiving off bioequivalent studies for generic formulation, not for new drugs, but for generic drugs, which I'll talk about in a minute. So here in class 1 drugs, they're very good solubility, very good permeability, no problem at all. They have good dissolution number, good absorption number. So they are ideal compounds. In fact, FDA, if the drug follows in class 1, if it is generic drug and immediate release dosage form, you don't need to do a in vivo bioequivalent study. You don't need to do a human clinical studies to compare the 2 formulations. Only for generic immediate release formulation. All you need to do is a dissolution study, in vitro dissolution study, and show the generic product as well as the innovative product are comparable within statistical limits. So they can grant a SDA bio waiver. So it means you save some money, but also you expedite the development of especially generic products. So this is one of the advantages. That is the main reason why biopharmaceutical classification system was developed. Now, they also good compounds for control these formulations because they are good solvent permeability. You can use this compound is ideal for control these formulations. Class 2, the problem is nothing with the permeability, but the drug is not very well soluble. That means you need to develop technologies to solubilize the compound. There are several technologies to improve, for example, salt formation, complex formation, nanosuspension, so on and so forth. Several compounds are made to the market in spite of being poorly soluble based on the technology used to improve the drug solubility. So there's a lot of scope for improving drug solubility. So again, the goal is to bring class 2 into class 1 by increasing drug solubility. Once you improve drug solubility, it becomes a class 1 drug. Class 3 drugs, of course, the problem is permeability. And majority of the drugs seem to be taken up by transporters. For example, acyclovir, for example. So you can design prodrugs like valcyclovir, which is a valine prodrug acyclovir, which is taken up by valine transport, amino acid transport of GA tract to increase absorption. So once you know what the challenge is, then you accordingly go back to the lab and design or make drug, change the structure of the drug or make formulation changes to make it bioavailable. So here the main challenge is permeability. So all the technologies are involved in improving the permeability by increasing the passive transport or even blocking the efflux transporters. Inhibiting efflux transporters or using transporters to increase drug absorption. Now, class 4 drugs are the most difficult one because you have two challenges, solubility and permeability. And it's also, I would call the nightmare because you have too many variables and too many things to control. Class 4 compounds are very difficult to formulate oral formulation. Either you have to find alternate routes or you have to develop technology which can solve both. There are a few technologies in the market. For example, self-emulsifying drug delivery systems. Here, use lipids to form an emulsion. The emulsion can actually not only increase the drug solubility, but also can increase permeability by the surfactants in the emulsion actually can act on membrane to increase permeability. Surfactant can also inhibit the PGP efflux, so they can also increase the absorption. So they can solve both solubility and permeability, but again, it is challenging because you need to use high level of surfactants, and there are some products in the market which use this technology to bring class 4 drugs in the market. So I said, you can use in vitro to predict in vivo. So if it's a class 1, you can actually develop a good in vivo correlation. So basically, you do a clinical trial first to establish that the dissolution studies correlate to the amount of percent drug absorbed. It's called point-to-point correlation. So in the future, you can use this prediction to any formulation changes. You don't need to go back to and do the clinical trials just based on the percent drug dissolved. You can predict the percent drug absorbed. So that is called in vitro in vivo correlation. So for class 1 drugs, you can expect good in vitro in vivo correlation. Class 2 drugs, not so much. Class 3 drugs, limited in vitro in vivo correlation. Of course, class 4 drugs, it's limited or no in vitro in vivo correlation. So because you can get a good correlation in vitro in vivo, you can get a biowaiver for generic drugs for class 1. And recently, FDA has also started giving biowaivers for a certain class 3 drugs with good in vitro in vivo correlation. So drugs, wherever you have good solubility, the FDA does give a biowaiver. But wherever you found low solubility, then you don't get biowaiver. So in other words, things which can predict it in vitro is much easier to get a biowaiver compared to the ones which cannot be predicted, especially for low permeable or low soluble drugs. So that was just on the physical property. Then Leslie Bennett in UCSF, University of California, UCLA, came up with, he found some issues with the BCS. He said, okay, permeability is good, but the problem is in many cases, you don't have you need to have the absorption data in order to predict the biopharmaceutical classification system, which is a lot more tedious and time consuming. So he used metabolism as a measure of permeability. In other words, the drug is highly permeable. That means it also can get to the hepatocytes and also be highly metabolized in both intestine and the liver. So instead of using permeability, use metabolism as a way to classify, a further refinement of the BCS classification, which is known as biopharmaceutical drug disposition classification system. Here, so what he predicted was the fact that this is primarily relies on the fact that a lot of the transporters involved in drug metabolism are both in the absorption as well as metabolism. So he used metabolism surrogate for extent absorption, which is easy to detect from in vitro hepatocytes test and then use it as a measure for classifying drugs. So what he found was, interestingly enough, most of the class 1 drugs and class 2 drugs are primarily excreted by metabolism. That means metabolism plays a significant part in their elimination from the body. Whereas class 3 and class 4 drugs, the metabolism doesn't play a major role, but renal or biliary excretion of unchanged drug. So class 3, class 4 drug primarily excreted unchanged in the urine or in the bile. Class 1 and class 2 drugs are primarily metabolized. So he used 70% metabolism as a measure of classifying drugs. Drugs which are show 70% greater are highly metabolized, which are less, they are class 1, class 3, or class 4. So he tried to refine that BCS because some of the BCS, because of the difference in methodology for finding of the permeability, I talked about how cathode permeability can be challenging sometimes, and same with rat permeability. Depending on what method is used, you can get different permeability and then you misclassify it, or have some drugs at multiple classification. For example, acyclovir. Some people are classified in class 1 until class 3. Same in bebendazole, class 2 and class 4, because of the method that is used for permeability. So, but you use BDS classification instead of permeability, you use metabolism, they fall in class 3, not in multiple classes. So they are less ambiguous. That, in other words, they are better refined models. So you avoid some of the misclassification or some of the limitations of BCS. So it's a refined model of BDCCS. In addition, the biopharmaceutical drug classification system can also tell you what are the effect of the transporters, what are the effect of drug-drug interaction, what is the effect of food-drug interaction. It's further refinement, not only just predict the drug disposition, but also transporter effects. So what he found was in the class 1 drugs, because mostly they're passively permeable, the transporter effects are minimal. Because they're highly permeable, the efflux transporters and the uptake transport do not play a major part. And they are not clinically important. Whereas in class 2, because they're poor solubility, the efflux transporter may predominate in their absorption. So you need to worry about class 2 drugs, you need to worry about efflux transporters. Class 3 drugs, because they're poor permeability, they're primarily taken up by absorptive transporters. So transporters are very critical. In class 4, both absorption and efflux transporters play a role in the absorption. So here he talks about further refinement, not only permeability, but what transport is involved, whether efflux or absorption. So class 2 efflux transporters, class 3 absorbed transporter, class 4 both. Class 1 is clean, as we talked about BCS. They are the ideal candidates. He also predicted the effect of food. So a lot of the drugs are given along with food. Food can either increase drug solubility, alter the metabolism, interact with the drug. They can also increase bile secretion and blood flow. So they can impact the drug absorption. So especially FDA would like to test in the presence of high fat meal in order to test the effect of food on the drug absorption. So what he found was class 1 drugs, if you give along with food, there is really not much effect on the absorption because the drug is already highly permeable. So food does not have very minimal role. The only the food will do is delay the GA transit time. As a result, increase the time to reach peak concentration. Does not affect the extent of absorption. Whereas for class 2 drugs, it is found that the food can increase the solubilization of the drug, or some cases some food substance can block the efflux transporters and increase absorption. So very important, they increase absorption. Food can increase absorb class 2 drugs. Whereas again, the transit time can be increased or decreased depending on the effect on solubility or on the GA transit time. And class 3 drugs, because absorption transport, here efflux is blocked, so that means more drug can go across the cell. In class 3, it is absorption transporters, right? So the food inhibits absorption transporter, the drug cannot be absorbed. So that's why it increases the drug absorption. Class 4, it can be mixed effect, depending on whether absorption transporters or efflux transporters play a significant role. So as you can see here, it not only can predict the transporter, but also the effect of food on the different transporters. So there's an advantage of BDCS compared to the other one. Another important thing is, as I said, remember earlier I said, it not only transporters, but where it is expressed, which side of the membrane, and whether it is intestine and liver. So just to give an example, the same transport expressed in the intestine and the liver can have different effects, depending on whether inhibit the intestine or liver enzymes or the transporters. So for example, if you have a drug which is a substitute of PGP, which is efflux transport in the intestine, also efflux transport in the liver it efflux into the bile or into the blood, whereas in the intestine it effluxes back into the intestine. This is CYP3A4 expressed both in the liver as well as the intestine. And here what it shows in this particular table is what is the effect of inhibiting the transporter or the enzyme or both on the extent of the drug that's available in the blood. So for example, you block PGP, more of the drug can get to the blood. Get in the blood, of course, can be metabolized, but more of the drug can get into the systemic circulation. So it increases. If you inhibit GP, it increases the extend absorption. But if we inhibit in the liver, remember here, so the liver takes up, and we'll talk about sometimes uptake transporters involved. We'll talk a little in a minute about uptake transporters, but assuming the drug goes in the blood, taken up in the hepatocytes, metabolized, okay? Now, if you inhibit this efflux transporter this side, the more of the drug is going to metabolize, right? So it will decrease the amount of drug available in systemic circulation because more of the drug is metabolized. If we inhibit the enzymes in the intestine, of course, it will still increase absorption because you're more of the unchanged drug can get into the blood. But if you increase metabolism, again, more of the unchanged drug. So here both are same effect, whereas the PGP has differential effect in the gut and the liver. If you inhibit both, because efflux as well as metabolism, you get much higher increased absorption. Both the challenges are taken care of here. But if you inhibit both, you have mixed effect, because if you inhibit metabolism as well as the efflux transporter, so it can increase or decrease depending on, or no effect depending on which is more predominant, whether the enzyme has more predominant effect or the transporter has more predominant effect. The point here is the same transporter or the enzyme can have a differential effect on two different tissues because of the way it's expressed and whether it's getting the blood or whether it's getting the intestinal lumen. So very important to understand as well. So the last model I want to talk about is a further refinement of BCS and BDCCS actually is the external clearance classification system. So this goes to a further step. They said it not only the route of elimination is important, but the mechanism of elimination is important, because this is again developed by Verma et al., who was a former NIPRite, and he has developed this. He again works for Pfizer. So he has developed this model in the recently 2015, of course 5 years now. So if you look at every 10 years, we are having BCS of 1995, 2005, you have the BDCS, and then another 10 years, you have the ECCS. So here they talk about rate of clearance is influenced by three things. Not only passive permeability, sometimes uptake transporters involved in the liver, and the extent of metabolism, and then finally, and also the uptake transporters in the urine as well as bile. So they consider all the mechanisms of elimination or clearance of the drug. So here is the blood. So the free drug, it can be actively get into the liver or passively get into the liver. Passive can go both directions. Active can only, or the transporter can go in one direction. It's influx or affluxed. Again, once it gets in the liver, it can actively get into the bile. So the clearance in the bile, and the metabolites can be cleared from the get out of the liver. In the urine, this is interesting. So you have the GFR, glomerular filtration. You have three mechanisms: active secretion, reabsorption, and the reabsorption in the distal tubule. So the drug can be filtered to the urine, through the glomerular filtration, and get into the urine. Depending on the drug's permeability, if it's highly lipophilic, it can again get reabsorbed. Most of the drug will get reabsorbed, not excreted. Now, some drugs can directly excrete from the blood into the kidney by active transporters, like penicillin, for example, get into the lumen, not by glomerular filtration, but active secretion into the lumen. Some can also be effluxed from the urine back into the blood. So there's a lot of uptake transporters, efflux transport in the kidney as well as in the liver. So this is much more refined than BDCCS. So BCS, then BDCS next step, ECCS and further next step, the mechanism. Here they did not consider solubility because that's not important in this particular case. It is more to use a predictive tool for early stage discovery to understand drug-drug interaction transporters as well as understand the mechanism of elimination. It is not used for biowaivers. BCS is used for biowaivers, BDC is used for finding out the elimination pathway, ECC is used to predict the mechanism of elimination of the drug from the body. They also use the PK of the drug, which is the ionization state, molecular weight, and log P are the important determinants of clearance. And I'll show in a minute why it's the case. So here, they found that class 1 drugs in the BCS, they don't use solubility here. They use the ionization constant. That means whether it's acids or zwitterions or base or neutrals. So highly permeable drugs in class 1, as you know, it's highly permeable. They divide two categories. Drugs with low molecular weight, more than 400 Dalton. Low molecular weight, primarily metabolism is the rate limiting step. That means they're able to passively go into the liver and get metabolized. Whereas if the molecular weight is higher, they cannot passively go through, but they are taken up by uptake transporters. So uptake transporters expression is very, very important. They're class 1B. In class 2, they did not find effect of molecular weight. Pretty much everything goes passively and heavily metabolized. Remember in BDCS, we said class 1, class 2 have extensively metabolized. Same thing is true here. But here, they classify based on the molecular weight, two different classes in class 1. In class 3, they're primarily hydrophilic drugs. They are not excreted as metabolites. If it's less than 400 Daltons, they passively, by glomerular filtration, excreted in the urine unchanged. Very important. Here, mainly metabolites is unchanged, class 3. And if it's more than 400 Dalton, they're taken up a transport in the liver, but they're not metabolized, predominantly metabolized. They excrete in the bile as unchanged and get into the feces. Or in some cases, they're taken up by transporters and get into the urine. Class 4, primarily passively, or some uptake transporters, renal excretion. They didn't find much bile here. So most of the class 2 and class 4 drugs are basic compounds and neutral compounds, whereas class 1 and 3 are acids and deuterions. So here, instead of use solubility, they use the ionizing constant, which is a much better predictor of the clearance mechanism. And this is the data. I will not go into the data. Basically, they talked about how well it's predicted, the model predicted the elimination pathway and the mechanisms. So basically what they came up with, here class 1 is primarily metabolism, but depending on the molecular weight, they will be passively absorbed into the liver or actively taken up by the liver. Class 2 is again primarily metabolites. Class 3 and class 4 are mainly unchanged drug in the urine or in the bile, depending on the molecular weight, whether class 3 or class 4. So how do they compare with each other? So as I said, class 1 based on permeability and solubility. Here class 2 is primarily on the extensive metabolism, whether a liver or kidney, that's what it talks about. Here specifically talks about transporters involvement. So this tells you the effect of food interaction, drug transport interaction. Here it goes further to predict the mechanism of interaction. Each one of them have a purpose. This is mainly used for biowaivers. This is used to guide new drug development, to understand the elimination pathway and also understand drug-drug interaction early on. This is further specifically look at the transport interactions and even changes in the expression transporters, how they impact the drug interaction or the drug transport into particular tissue. So this is basically, you see a kind of overview we talked about. So BCS is mainly used for biowaivers. The other two are mainly used for drug discovery and development because they build on BCS, BDCS, ECCS builds on both the models to predict better the clearance mechanism. So ultimately the goal, if you look at all of these approaches, to make a drug bioavailable, bring all of the drugs into class 1. If you make a class 2 drug, you increase solubility, become class 1. If you improve the permeability, become class 1. Improve both solubility and permeability, again become class 1. So that is what here is mostly formulation optimization. The class 4 and class 3 will require structural changes, lead optimization to bring it in class 1. So you can, depending on which class of drug, once you know the solubility early on by high throughput screening, again permeability based on CACO2 cells, you can predict early on whether the drug is going to fall in one of these classes, and then accordingly develop strategy to improve the drug properties and optimize the lead to have better success in the later stage of drug development. So you wanted to predict early on, not after that, what I talked about earlier, in the initial part of the presentation. So with that, I want to come to the summary slide. So this is the development NCs or marketed drugs. So you have combinatorial chemistry, high throughput screening, biotechnology, and molecular modeling. All of them are guiding the new chemical entity development. Whereas in market drugs are developed by patent expiry, market forces, therapeutic potential, clinical need. But the better drugs are better delivery systems. You take a market drug, make modifications, and develop as a new drug. So you can use this as a guiding tool, the BCS, ECS, ECN, and BDCS, and you can develop an oral deliverable compound. If it doesn't, you can also develop new drug technology, or you can find alternative routes if oral is not possible. So it's a good roadmap to use biopharmaceutics to guide drug discovery development to ensure successful transfer of the molecule from the early stage to the later stage of drug development. So with that, I'll stop here, and I think I went a little bit over, but hopefully it's helpful. So I'll stop here and take any questions. Thank you for your attention. So Dr. Ramani, hope I didn't go overboard on time. A little bit slightly over.
Yeah, but it's all right. It's very informative. I think I like it. Yeah.
Thank you very much, sir. It's a very nice and informative session. Sir, let us start the question and answer session.
Sure.
Sir, one of the student is asking, his question is, apart from polymeric nanoparticles to the brain, can we formulate using apolipoproteins to deliver the drug to the brain?
Sure. Yeah, good question. I'm assuming somebody must be a pharmacy student asked the question. So it looks like so.
Yes, sir.
Yeah, so you're talking about what you're talking about is a target delivery. So you're talking about attaching a target ligand, apolipoprotein. In fact, there have been a lot of approaches. People use apolipoprotein to target the brain. That is one of the ways to target, but you don't. There is even I believe there is a I wouldn't say clinical trial. There's early stage preclinical development going on using apolipoprotein for the brain delivery. There are a lot of even transferrin, which is an iron transferring protein, has been extensively used to target drugs to the brain. Yes, to answer your question, can apolipoprotein be used? Yes, it can be used. Of course, you need to understand it's a large molecule. So again, you need to conjugate to the nanoparticles or coat. Some people have coated the apolipoprotein to target the drugs better in the brain. So the problem of the blood-brain barrier, and although people talk about targeting, the question is how much dose are you able to deliver, right? So it depends on the expression of the apolipoprotein. And remember, most of the time we test in a normal animal model, but in disease model, especially if we're talking about CNS disorders, the blood-brain barrier changes as well as the expression of the protein changes. So you need to take that into account. Often what I see is people do in a normal animal said, okay, everything goes well, but then they really take it to the next level. It doesn't work as well, or sometimes it works better. We get maybe leak here in inflammatory conditions in the brain, and you may or may not need the apolipoprotein, may still be passively able to go through. So it is important to understand the expression level and also test an actual animal model, which predicts the distribution in that particular disease model as opposed to just predicting based on a normal animal model.
Sir, another question by the same participant. So he's our research scholar, Mr. Manish, in our pharmaceutics department.
Okay.
So he's asking polymeric or lipid nanoparticles, which one is best for delivering the drugs to the brain?
Okay, so
because he has a concern over polymeric nanoparticle toxicity. He's working on the delivery of the drug to the brain. He's working on the project.
Sure. So, I mean, so nanoparticles, of course, you can make by different materials, right? So you can make polymer, you can make inorganic materials, you can make lipid, you can use proteins. So a whole set of lot of research has been done on this, right? So it's not a question of which one is better. In one way, the question is good because it's always a question, okay, you have multiple things to choose from. So which one do you choose? So there's really not much out there in terms of comparative studies, but now, to answer the toxicity question before I talk about the comparison, the toxicity all comes down to the material toxicity, right? So if the material inherently is safe, it does not matter whether you use lipid or polymers. So I don't want to assume that all polymers are bad. In fact, you have a lot more polymers like synthetic polymers in the market, which are approved for different various conditions. So I don't want to say all polymers are toxic. Of course, anything for toxicity doesn't matter with polymer or lipids. Even some lipids have been found to be toxic. So the question is, it's more not necessarily whether polymer is toxic, polymeric materials are toxic, lipid. It's the inherent toxicity of the material per se, which dictates toxicity of the nanoparticles. Now, as far as comparison is concerned, there is really very less information in the literature because some people say polymeric particles have gone in a particular model and then lipid nanoparticles. So it's important from product development, it is important to compare head-to-head comparison and see what serves the particular condition. So it depends on a lot of different things: drug loading, drug release, premature drug release, as well as the size of the particles, right? And what surface modification you can do versus you cannot do. So there are pros and cons for each one of those approaches. So again, the question, again, I always go back to the 4D. So to ask the important question, not to develop something and retroactively look at drugs, rather right from the beginning, ask the question about the disease, the drug, destination, and according choose an appropriate material which will suit the purpose, opposed to just saying, okay, I have this lipid, let me load the drug and retroactively deliver the drug. So it's always good to start from the other way around, starting with the 3Ds before to the delivery. So from toxicity, it doesn't matter. It depends on the inherent toxicity of the material per se, not necessarily the polymer nanoparticle, but the toxicity of the material per se.
Thank you, sir. Sir, another question from one of my students. So they are sending all the questions to my WhatsApp. How about use of aerosolized nanocarriers for lung-targeted drug delivery in case of COPD or else present ongoing pandemic corona? So this question is by Kirtana.
Excellent question. So, in fact, already if you're watching the news, there is already remdesivir, which is an antiviral drug. So recently there was the Gilead Pharmaceutical started a I think the formulation is in clinical trial on the aerosolized inhalation for COVID. So COPD, absolutely. In fact, there are a lot of products out there. Tobramycin, for example, antibiotics delivered as inhalation for COPD, chronic obstructive pulmonary disease. Now, as far as the COVID, right now we don't have anything. That's a good approach, I would say, especially to treat the inflammatory condition, especially in the lungs, right? Early stage of the disease, because now we know that COVID not only impacts lungs but also the other organs as well. But maybe early stages, it may be a good approach to deliver some of the antivirals. And that's, as I said, Gilead Pharmaceuticals currently looking at a clinical trial, developing an inhalation formulation. Right now, remdesivir, I think for emergency approach is given as intravenous. So now they're trying to develop a aerosolized formulation. So that's a good approach. I would say looking at some antivirals as well as COVID-19 would be an approach. I'm sure that some of the companies are looking at it. Probably the data is not out there. That's why probably you don't see in public domain. Yes, it is a good approach for localized disease.
Thank you, sir. Sir, another question from my fellow faculty. She is Kalyani, and she is asking on what sort of neuronal cells like CaCO2 cells for adenocarcinoma, the biocompatibility can be tested in vitro biocompatibility testing.
So I'm assuming this is, so they're talking about testing the drug in a tumor for cancer drug targeting or general toxicity testing. I'm not sure what the question is about, but I'll try my best to answer the question. So
she's asking about neuronal cells and what sort of neuronal cells like CaCO2 for adenocarcinoma. We generally use CaCO2 for adenocarcinoma. So she's asking which neuronal cell is suitable for biocompatibility testing.
So you're talking about inorganic nanoparticles, I'm assuming CaSO2. Is that the one? Cerium.
Sir, excuse me, sir.
Yeah. Please go ahead. Yeah.
What I'm asking is we are testing the for adenocarcinoma we are testing on cacao2 cells.
Okay,
so in the similar manner for testing the neuronal cells biocompatibility, what cells can be used?
Oh, I see. Okay. So I specifically, I don't know about the neuronal cells per se, but what I'll tell you is, okay, the reason we're using colon carcinoma is for a different reason. Actually, for toxicity, general toxicity studies, generally they use it as a primary cell line, generally speaking, which means you have to isolate not a tumor cell. Now, cacao2 cell is widely used. Of course, you can use both depending on type of condition you're trying to treat or what your question is in terms of the toxicity versus the permeability. Now, cacao2 cell is an immortalized cell line. In other words, because one of the challenges primary cell line is if you don't maintain properly, it gets contaminated and it's very difficult to maintain the cell line. So when you immortalize, it means if your tumor cell keeps multiplying, it's easy to maintain those. So neuronal cells, typically what people use, more than neuronal cells, I think it depends on specifically what you want to test. And if it's only purely toxicity standpoint, if you're looking at permeability, people use rat brain endothelial cells. They are primary cell lines. They're very difficult to maintain. So that can be used. Some people use glial cells to test some of the toxicity studies. Now, again, depends on if it delivers to the brain. And I would basically start with the RBEC, which is a rat brain endothelial cells, to test the toxicity, general toxicity. If they're able to deliver the drug, but you want to make sure the nanoparticle per se is not toxic, that's the cell I would use if your goal is to deliver to the brain. But if you general cell toxicity studies, people generally use the monkey kidney cells is one of the cells that's very commonly used. General toxicity for cells, cacao2 cells. Technically, it's depending on again what the question you're trying to address in terms of toxicity versus delivering to the carcinoma cells. I would say cacao2 cells can be used. You can do MTTSA to understand cell viability, but it's not a true indicator of cell toxicity because it's already adenocarcinoma cell line, right? So I would use a primary cell line to test toxicity, but brain, I would start with brain endothelial cells.
Okay, so thank you so much for.
Sure.
Thank you so much.
Sir, I have a question from the YouTube live chat. So the question is by Pramila.
Mhm.
So what is the biopharmaceutics role in protein engineering?
Okay.
Role of biopharmaceutics in protein engineering.
Excellent question. So I didn't cover that. So most of my discussion was based on small molecules. So that is a lot more challenging, right? So biopharmaceutics, of course, one you need to think about biopharmaceutics. We don't have any orally delivered protein, right? The only protein that is delivered other than injection is the insulin, inhalation insulin. That's the only developed as a non-injectable route. Everything is injectable, right? There's no protein currently. It's delivered orally or even any other um, alternate routes other than injection, subcutaneous or intramuscular. Those are the common routes in administration. The reason biopharmaceuticals is a lot more complicated there because, of course, they don't get absorbed very well across the membrane. They're macromolecules, as we know, right? So challenge a lot more. It is not as simple as doing a small molecule. So right now the most of the development focus is on how to develop a stability is a big issue. Chemical, physical, and biological stability is the biggest challenge in biopharmaceuticals. We're talking especially protein drugs. So the challenges are not solubility is not a challenge, right? Most of the protein drugs are hydrophilic and soluble. Unless they aggregate, they can reduce solubility. Permeability is a big challenge. They cannot cross the membrane. So there have been some approaches delivered, for example, insulin by oral route, using some enzyme inhibitors to prevent the degradation of the protein in the GI tract, and also some absorption enhancers to increase the permeability across the oral route. Um, Somewhat successful, of course, and still a long ways to go. So typical things that would apply in a small molecule will not apply to a biopharmaceutics. Solubility is not an issue. So the challenges are more, the goal in formulation biopharmaceutical is mostly the stability rather than, at least now, rather than delivery. So it will take a long time. I mean, if we can get anything delivered orally. The only closest I can think of insulin is all delivered by inhalation, right? Because of the porous structure of the lung as well as the large surface of the lung. So you're able to deliver insulin through the inhalation. There is also a formulation, I think, is a buccal spray. People use to avoid some of the enzymatic breakdown, as well as the literally better permission than the oral route. So to answer your question, it is a lot more challenging, and the challenges are very different than small molecules.
Thank you, sir. Another question from YouTube live chat. So this is by Navyat, third year BPharmacy student. So he's asking which approach is most effective to develop formulations for class 2 and 3 drugs to increase the solubility and all.
Very good question. So what is the most easiest approach? So, I mean, I didn't get a chance to, discuss the whole addition chart. So in class 2, of course, solubility is an issue, right? So there are multiple approaches. You can do salt, you can do complex formation, you can do nanosuspension, you can do emulsions. So the question that you'd ask, if I can go back to the slide. I mean, I didn't get a chance to talk about the decision tree. So you could actually use the decision tree as a starting point to, I don't know if they can see me. They probably cannot see in the YouTube live. I don't know. Can they still see my slides?
Yes, sir, they can see.
Okay. Let me go to slide then. Okay, here, this is the slide. Can you see the slide?
Yes, sir.
Okay. So if we look at this, so depending on for example, first you say, start with, first if it's an ionic compound, can a salt be made, right? You make a salt, it's easy to make a salt and water soluble, but you just adjust the pH. It's a little easier approach. If the log P is low or high, so if the high log P and the melting point is again low or high, depending on that, you can make a, co-solvents or micellar dissolution or emulsions. And if it's based on the crystallinity of the compound, you can make it amorphous and make a nanosuspension. Again, you can, depending on the chemical structure, physical properties, you will start the simplest approach to the most complex. You don't directly go to the emulsions or nanoparticle nanosuspension. If you can, the most common approach is easiest and most common approach, because majority 70% of the compounds in the market are weak bases. That means you can form hydrochloride salt. That is why hydrochloride salt is the most commonly used product in the market, because you can easily make a salt. So if salt can be made, that's probably the easiest approach. If that doesn't work, then you can go to more complex approach like nanosuspension and emulsion. So you have to understand the question about, ask the question in terms of whether it is crystalline, non-crystalline, whether it's ionic or non-ionic. So accordingly, then you decide which approach to use. As far as class 3, again, the most, I would say the most effective approach, for example, I talked about acyclovir. So acyclovir has poor passive permeation through the biological membrane. So what people did was they actually attached valine with an amino acid to acyclovir to form valcyclovir, which is bioavailable because it's better absorption because it's taken up by transporters. So acyclovir is still in the market, but valcyclovir has a higher bioavailability because valcyclovir is taken up by transporters. So basically, you attach a prodrug moiety to match the structural features required for a transporter. As a result, you can enhance absorption. So that is the most effective approach. Of course, that means that you have to change the structure. Another approach is you can also block the PGP efflux. If you know PGP efflux is the one that's preventing the absorption, then you can inhibit by using co-administering another drug, or for example, as simple as piperine has been shown to inhibit PGP, or CYP3A4 intestinal enzymes to increase absorption. The third approach would be using absorption enhancers. They've not been very effective, but you can actually use, for example, to open the tight junctions transiently, or in some cases you can use the microemulsions to use lipids, and then they're taken up by a process called chylomicrons. The same way the bile acid transported into the body. That's the third approach. So it'll all depend on what level of absorption is required, and whether, of course, you can formulation approach much easier than making a prodrug, because then once you change structure, you have to go through all the tox studies, safety studies, and PK studies all over again. So you can start a simple formulation approaches to increase the permeation across the membrane, and or using formulation to like lipid-based formulations to for the drug to be taken up by the lymphatics, which is lymphatic supply using the bile to increase the drug absorption. So those are some of the approaches.
Thank you very much, sir. It's a very enlightening session. Sir, Kumar sir.
Yeah.
So, over to you for thanks note.
Dr. Perumal, if you are okay, it seems there are 2 more questions running up.
Sure, absolutely. Please go ahead. No, I'm fine.
Yeah, Sunil, you may ask those 2 questions as well.
Yes, sir.
Because they keep waiting. Even after 2 hours of session, people are interested in asking questions, so we should answer them.
Yes, sir. Sir, this question is by Akila Ravikumar. Sir, is asking if class 2 drugs by change in particle size when formulated, can they be further subclassified?
I'm not sure what the purpose of subclassification, right? So your goal is to increase solubility, right? Now think about why they classify. They want to make it simpler, not more complex, right? The simpler, the better. The more subclassifying, the more challenging it is. Of course, biology is a lot more complex than just classifying the drugs. So here the goal in class 2 is based on solubility and permeability. Again, they define what is solubility based on solubility in pH 1 to 7.5, and also permeability more than 90% absorption. So particles reduction is a good way. Actually, it's a very easy and simpler approach using amorphous from a crystalline to reduce the size. It'll increase surface area and increase the drug solubility. As a result, increase the drug absorption. So you don't classify drugs based on the technology, but you classify based on the drug properties, not the formulation properties. Hope that answers the question.
Yes, sir. Sir, another question by Harish Chandra, a 4th year B.Pharm student. He's asking where the industries and institutions, education institutions are lacking in the sharing of the knowledge, especially in the way of research.
Wow, I mean, it's a very thoughtful, excellent question from a B.Pharm student. That's a wonderful question. I'm really impressed by the level of engagement of the undergraduate, especially. So what is lacking, right? I mean, that's why we are all in the business, right? We are all doing research. Now, as I said, in academia, of course, our goal is to disseminate the knowledge, publish, and share the information. Of course, today's webinar is an example of that, right? So sharing the information. Industry, Dr. Namani can share more than I can. Of course, I have very limited experience in industry, but the biggest challenge in industry, of course, there is a reason they are there for business, right? So they don't want competitors to know what's going on. Typically, if things are published from industry, that means things didn't work. Or there is maybe they want to change the opinion or policy decision. For example, they talked about ECCS or Lipin's Rule of 5. So why did they publish? Because one, of course, definitely have societal needs in mind, and they want a broader knowledge, a broader input. It also change thinking of the regulatory agencies when it's validated, more publicly validated by other companies. Of course, very rarely they'll, of course, disclose very specific about drugs if they are developed. And after the developed clinical trial, those things will, of course, will come out. Of course, if they're encouraged to validate their, the product or what they try and develop. So, but in early stages, they are not ready to disclose much information public because obviously proprietary reasons as well as for competition reasons, they would not. So that is where the difference is. I wouldn't say lacking, it's more of, but it's changing actually. I don't know how much is happening in India at this point, but what I'll tell you is, if you have heard about open innovation, it's a known concept in IT world. It's happening in all the big 10 pharma companies now have open platforms. For example, SDC has an agreement with Eli Lilly. So what is happening is, if you look at the industry landscape, they're shrinking, they are merging, they're buying, so what they're trying to do is leverage resources from academia, best of both worlds, right? Academia has a lot of good ideas and industry has a lot of resources. They're trying to match that. So what LL is, for example, Pfizer, all the big companies have this open innovation. So if you have a good compound, you found exciting data, they will have an agreement, they'll screen for free on multiple targets. Of course, free in the initial stages. If they find something interesting, they will come back to you and have a specific agreement to take it further for clinical development. Of course, assuming that you have covered the patent and all those things, because patent is very critical for industry. They don't want to invest in technology which is half patent. And that is one of the biggest, right now in the United States, there is a debate. I mean, for example, hydroxychloroquine, because off patent drug, right? Companies are not interested. So there is a little bit of, again, I'm not taking sides here, but there is a debate. Academia wants to push it, but in some industry, of course, there are some pros and cons of hydroxychloroquine. Industry is concerned because it's such a cheap drug, then that will hamper the development of new drugs. So there's a lot of business reasons for doing it. So lack of information is, I think that is changing. It will definitely change because at some point, of course, still is going to be limited from industry standpoint what they can share. But I think that the tide is changing. So you'll see best of both worlds coming together. But at least in the United States, some universities are doing better than others, but some universities have a long ways to go because the relationship has to be built on a systemic way because, of course, there's always trust building is part of the challenge in terms of academia and industry. Time is a big factor in industry. Academia, you have all the time in the world. Industry is very time-bound, product-oriented. You need to get everything in place. I'll just share one example, then turn it over to Dr. Namani. I'm sure he can share more than what I can from industry. When I was in Ranbaxy in the early stages of my career, one of the director told me this. He said, academic research is boiling water in an open vessel. Industry research is boiling water in a closed vessel. So hopefully that sums up what I wanted to say.
Yeah. Sunil,
Yes, sir.
so why don't you answer the last question by the Rizwan also? Not understood that question, but
In the live chat, sir.
yeah.
Muhammad Rizwan.
This will be the last question, Dr. Pillai.
Sir,
No problem.
he's
I'm glad
asking,
people ask questions.
there are a lot of techniques to enhance the solubility. So one of the method is solid dispersion technique. So if the solid dispersion method is if it could give expected solubility, so why there are so many methods? This question is like that.
Right. So again, go back to the decision tree, right? So it's not one size fits all. If there's only one method, we'll all be out of business, right? We'll all be sitting at home because no nanoparticles, no nanosuspension, no emulsion. No, the serious answer is it depends on drug properties. Remember I said, you cannot retrofit, right? It's not one size fits all. If you cannot have one technology, because there's so many technology, because the current technology cannot adapt to this other drugs, right? So it depends on drugs' physical properties. Yes, solid dispersion is a good technology. Now, the question is how much solubility enhancement you can have and how much polymer you have to use. You can get, let's say, very good solubility, but you have to use a gram of polymer. Your drug is only, let's say, 20 milligram. So that technology is not feasible because your drug is so much, and sometimes the formulation issues come up and compressing all of the other formulation factors come in. So it depends on what do you want to accomplish, whether it's purely solubility alone or solubility and permeability or what extent of solubility, right? So, and whether even in spite of forming solid dispersion, are you still able to achieve the target solubility? So solid dispersion is not a one size fits all. And also the fact that, there are different types of solid dispersion. If you go into the details of solid dispersion, there are complexation, there's also co-salt formation, co-crystal formation, so a lot of different approaches. So again, it comes back to the properties of the drug to choose the appropriate technology, not the technology and retrofitting the drug, but starting with the drug and seeing what is the right fit for that particular drug.
Yes, sir. Sir, my colleague, Dr. Ravi Kumar, is asking one question. To enhance the bioavailability, the methods described in the presentations can apply for ophthalmic drug delivery for making nanoformulation. So he's working on ophthalmic nanoparticles. So he's asking whether the decision tree is applicable for that ophthalmic nanoformulations also.
I mean, broadly, yes, but I think that is specifically designed, I would say, mainly oral and injectable routes. Ophthalmic has a little bit of different challenges. The volume of fluid is very limited, and corneal permeation is also a little bit challenging. It's more of retention, more the deliveries, how long can you retain in the eye, right? So yes, as a matter of fact, yes, you could use nano suspensions. You could use same technology, but of course, some modifications, right? So again, the amount of drug that you can incorporate in ocular has to be very, very limited because it will cause irritation in spite of being a nano suspension. So the answer is broadly yes, but it has to be customized to the route administration. That decision tree broadly applies to solubilization, but the question is, given the dose and the route, need to be a little modification may be required. So, but that decision tree is primarily, that was of course coming from one of the companies. They're primarily focused on developing the technology mainly for, I would say, parental injection as well as oral. But having said that, you could apply that, but with the consideration of the volume and the route administration.
Sir, there is another question, if you don't mind.
Sure, go ahead, please.
So this is by MPharm student Kirtana. Okay, so see, she is interested in magnetothermal drug delivery. She is asking whether this magnetic field will work out perfectly for targeting the tumor.
Yeah, I mean, this is much broader question, right? So what you're talking about is physical physical stimuli for drug delivery, right? So yes, there have been some approaches. Clinically, I mean, people have even used magnetic force for like using inox and nanoparticles. They have been, this is my personal opinion, but the more complicated the technology, the challenge, there's a lot more challenge in bringing it to the field, right? So when you think about FDA approval, you're not only talking about drug approval, you're talking about device approval. So it comes in a totally different category of drugs. So they also call this a combination drugs, combination products. And once you have both device and a drug product, does it work? Some preclinical studies have shown there's some clinical studies. But again, depending on what are the advantages of existing technology, that would be the question to be addressed. If it's a difficult to treat tumor, yes, people will probably go for it. But again, application is going to be very limited. You cannot, again, not a one size fits all. The answer is there have been some studies, inox and nanoparticles, they're able to use. And in the context, I think there's a lot more interest in what is called as ultrasound-based microbubbles. That's where you can apply ultrasound using microbubbles, targeting had a lot more attention than the magnetotherapy. But I think, again, there's still long ways to go. So it's very early to say whether it's going to be clinically feasible or not, right? So certain tumors probably it'll apply, but I don't think it'll broadly apply across the board. Certain difficult to reach tumors, yes, maybe it'll apply. And again, the question is it able to achieve more or better than what we currently have? That's the question need to be addressed.
Sorry, sir. There is another question from Kartaya University faculty. So Usha Kiranmai.
Sure.
So she is asking about dose number. Simply, can you explain it again?
Yeah, dose number is okay. Dose number is, let's say 100 million of the drug. Okay, 100 million of the drug. So the volume of the fluid, the intestinal fluid is 250 ml. Let's say 250 million of the drugs to make it easier from math standpoint. 250 million of the drug in 250 ml. Right? So, and then the solubility is, let's say, 10 milligram per ml. Right? So basically you get the numerator. So if the numerator dose volume, that means if you're able to solubilize all of the dose in the 250 ml of water, 250 ml of the pH 6.5, I should say specifically intestine, right? It means your solubility, what you get, so when you divide the dose by the volume, you get, actually what you get is solubility, right? 250 milligram, you basically 1 milligram per ml. And with drug soluble 10 milligram per ml, definitely you're below the saturation concentration. That means the dose is soluble, so it will not cause any issue, solubility issue in vivo. On the other hand, if your dose volume comes out to be, let's say 25 milligram per ml, and your drug solubility in the denominator is 10 milligram per ml, of course you're exceeding the solubility. That means it's not going to be, the dose is not going to completely soluble in the in vivo. So dose volume is basically telling you the same thing as solubility. It's telling you solubility of the dose in the total volume of the intestinal fluid. It's the same thing telling in a different way. So it is, in other words, saying that, okay, I know the solubility of the drug is 1 milligram per ml. So what does it mean? Can I deliver 250 milligram the dose? The question can be only answered if you have 250 milligram divided by the volume of the intestinal fluid, and if that value comes out to be lower than the solubility, it is good. If it is higher than the solubility, then it is not good. That is why I said the dose volume dose number should be lower, not higher, because the value, one is numerator, the other one is denominator. Hope that clarifies.
Yes, sir. Thank you, sir. Thank you very much for enlightening us. There's a lot of things that personally I have learned, and most of the students have interacted with me over the WhatsApp itself for asking the questions.
Thank you. I'm glad it was helpful. Thank you very much.
Sir, over to you, sir. Kumar, sir.
Yeah, thank you, Dr. Pillai. There are three phenomena which I observed. I think this is the first time ever in this webinar series of SVCP this much long the webinar went on, and the way the questions are coming, they keep on coming, and we have to in fact stop asking questions. So there is definitely more questions than answers. That is how the science progresses. I know about Dr. Pillai, and he can talk continuously, non-stop, and he can ask any number of questions. He can answer like Rajnikant with one shot with 100 thing. So that is what is Dr. Pillai is. He's my opposite roommate, so he knows more about me, I knows more about him. So we'll talk it on a separate platform on that aspect. But I saw Dr. Pillai as a friend so far, but now I'm saying with my lens as more than my friend, as a Professor Pillai. Another P added to you now, when the way I look at you. You're able to maintain the tempo, and people are sitting even after the sunlight is gone here, and watching your presentation. I think it tells a lot to me. I am personally said that the phenomena, first phenomena is the way you present, the way you answer. And the second phenomenon which I got really is excited is even the BPharm third year students are asking very, very relevant and interesting questions, starting with Navyat and then moving to